Today's lecture is the first in a series of lectures that is an entire unit dealing specifically with the adaptive immune response. More specifically, this lecture is part of two lectures that focuses in general mechanisms of lymphocytes as well as some more specific detail with B cells. So just for a quick review, what we focused on in the previous unit of material was getting a general understanding of the cell types involved in the overall immune response, as well as the organs involved in the immune response. And then we spent some time talking about the innate immune response. So as I just mentioned, we are going to focus now on the adaptive immune response. And just to refresh our memories a little bit, I want to highlight a couple of very important features or characteristics of the adaptive immune response. With adaptive immunity, this response typically occurs within days of an infection. So there is a time lag associated with the adaptive immune response kicking in. This is in contrast to what we heard or learned about the innate immune response, where that is an inborn mechanism that occurs within minutes to hours of an infection. Adaptive immunity also focuses on highly specific mechanisms, meaning that the detection occurs with an extreme amount of specificity down to the species level. This is in contrast to what was happening in the innate immune response, where very broad molecular patterns of pathogens were being detected. The adaptive immune response is also referred to as being very diverse. It is diverse because there are a large number of diverse and distinct receptors that are involved in the recognition that's going to occur for the specific pathogen. So the specificity goes hand in hand with the diversity. The specificity allows us to identify a specific pathogen, whereas the diversity of the receptors is what allows for that specific detection to take place. This is in contrast to the innate immune response, where in comparison there is a much lower or limited number of receptors involved in the recognition process. In addition, the adaptive immune response has what we refer to as a memory component. It is a persistent memory, meaning that once an infection has taken place and an immune response has been elicited against a specific pathogen, there are memory cells that are produced that allow for a very quick response when a subsequent infection takes place. This is in stark contrast to the innate immune response, where there is no memory component, no recognition of previous pathogens that have been detected. And so again, this is a complete difference between the adaptive and the innate immune response. With adaptive immunity, the major cell types involved are going to be lymphocytes, particular B cells and T cells, but also antigen-presenting cells are very important in this mechanism. In comparison, in the innate immune response, we know that phagocytes were the number one category or type of cell that play a role in innate immunity, including neutrophils and macrophages being the top two phagocytes involved. Also, natural killer cells are typically involved in the innate immune response, and dendritic cells do play an important role. Now, as I mentioned in a previous lecture, it doesn't mean that only certain cell types are involved in certain mechanisms, but there are cells that are dominant in each one of these types of responses. This portion of the lecture is going to focus on antigens and antibodies and T-cell receptors, so we need to examine some basic definitions before we continue on. We've already established the definition for the term antigen, but again, I want to refresh our memories because we're going to focus on this for today's lecture. An antigen is any substance that binds specifically to an antibody, which is a receptor found associated with B cells, or a T cell receptor known as a TCR. So an antigen is a very general term, but is defined as something that can bind specifically to an antibody or a T cell receptor. Now, an antibody is a protein that is also referred to more specifically as an immunoglobulin, and this is a structure, a molecular receptor that recognizes a specific region or component of an antigen. Antibodies are specifically associated with B cells, and as we mentioned in a previous lecture, come in two different types. There are membrane-associated antibodies, as well as antibodies that are considered soluble and are secreted from the B cell, and we will look at that in a little more detail. But what I do want to reiterate is another concept that we talked about in a previous lecture, was that for B cells and for T cells, they are found in what's known as a naive state. In the naive state, these cells have not interacted with any antigen just yet and require activation or interaction with an antigen to occur. When that happens, there is a complex process of differentiation and development that occurs for that lymphocyte. Now, for B cells, because we're talking about antibodies in this particular case, antibodies are specific to B cells. For naive B cells, the antibody is in fact membrane bound. We also find membrane associated or membrane bound antibodies with memory cells. And the activated cells, which are also referred to as plasma cells, that are produced once the activation, that first interaction with an antigen takes place. These activated B cells are the ones that produce a soluble antibody, one that can be secreted from the B cell. So again, two categories of antibodies, membrane-associated or membrane-bound versus soluble. And the cell types that are associated with either one of those, predominantly naive B cells as well as memory B cells, will have the membrane-associated form, and activated B cells are going to produce the soluble or secreted antibody. Immune cells, that is, lymphocytes, B cells, and T cells, do not interact or react with the entire immunogen component or the entire antigen. In fact, what happens is there's a specific interaction or association at discrete sites. These discrete sites are known as epitopes. So an epitope is a portion of an antigen that reacts with the antibody or the T cell receptor and is also referred to as the antigenic determinant. So this is the portion of the antigen that specifically binds to the antibody or the T cell receptor. So in this particular slide, you have a figure where on the left-hand side you see an antibody, and at this point it doesn't really matter if it's a soluble antibody or a membrane-associated antibody, but what this figure really nicely does is hone in or highlight the portion of the antigen where that direct interaction is taking place. And in this diagram, it looks just like a key. So the portion of the key that you hold, or the entire key I should say, would be considered the antigen, and the ridges or the specific part of the key that go into a typical lock would be the epitope. So this is the specific part where that interaction, that recognition is taking place between the antigen and the receptor. This holds true for T cells as well, in that the entire antigen is not typically detected. It is usually a portion of the antigen where the interaction takes place, here too referred to as the epitope. However, on this slide, it's also a really good point for me to introduce the complex interaction that takes place between antigen and T cells. In this particular figure, we're looking at helper T cells, but the same holds true for cytotoxic T cells as well as regulatory T cells. T cells cannot bind directly to an antigen. Let me repeat that. T cells cannot bind directly to an antigen or an epitope of an antigen. There needs to be a very complex interaction between a cell category known as an antigen presenting cell. So we introduced this term APC, or antigen presenting cell, a lecture or two ago when we discussed different cell types. In particular, dendritic cells are very common antigen presenting cells. Their job is to do exactly what the name implies, is present antigen to T cells. And it is a very complex interaction like you see here on the slide. So we're looking at the right-hand panel. The yellow cell represents an antigen presenting cell, and there is a receptor that does the presentation of the antigen, which is represented by the little red dot or sphere, and that is then presented to the T cell, in this case a helper T cell. So the helper T cell has its TCR, T cell receptor, and there's this complex interaction between T cell receptor, antigen, and presentation by the antigen presenting cell. So again, T cells cannot bind directly to an antigen. They must always have antigen presented to them by a broad category of cells known as an antigen presenting cell. And this complex interaction is that direct link between the innate immune response and the adaptive immune response. Phagocytes in the innate immune response phagocytose or break down the potential pathogen. And we discussed the fact that there are two fates that are associated with those breakdown products. One fate was to just be released from the phagocyte, and eventually those components are released from the host system. But the alternative pathway is for those pieces and parts that have been produced by the degradation of the pathogen via phagocytosis to then be utilized in presentation to T cells. And that again is that link between the innate and the adaptive immune response. T cells again cannot recognize or interact with antigen on their own. They must be presented to them by an antigen presenting cell. Now the epitopes that associate or interact with the B cell receptor or the antibody or the T cell receptor associated with the T cell is going to be a little bit different because of the mechanisms involved in allowing for the interaction between antigen and lymphocyte to occur. So for B cells, soluble antigens can in fact be detected. This includes an intact pathogen with a portion of the pathogen representing the epitope. It can also be something that a pathogen secretes. We mentioned at least once before that some pathogens produce toxins to help with their infectious process, and these are things that are secreted by the pathogen and enter into the host system. Those are also molecules that can interact with an antibody or the B cell receptor. T cells, however, in contrast, can only have or recognize antigens that are bound to another receptor. That receptor is referred to as MHC, which is short for major histocompatibility complex. And again, we will discuss that in more detail when we get to T cells. But we just focused a minute ago on that complex interaction that needs to take place. So B cells can interact with soluble antigens. T cells can only interact with those that are presented to them. B cells can interact with very highly accessible exposed surfaces of a pathogen. T cells, in contrast, can only interact with something that is derived from an enzymatic digestion or breakdown of some sort. For B cells, oftentimes the sequence of amino acids can either be linear that are recognized or conformational. This is because the B cell receptor or the antibody can bind to an intact pathogen that still has conformation, three-dimensional shape associated with it. T cells, because the only way they can interact with the antigen is for those components to be broken down, will not have recognition of any three-dimensional or conformational components. So for B cells, what can be recognized is something conformational, three-dimensional, or can also be a sequence of amino acids, a linear stretch of amino acids. For T cells, there is no conformational component that would be recognized. It is only going to interact with linear sequences of amino acids. So what we've just heard or discussed was the fact that B cells and T cells interact with antigen in very different ways. What I want to discuss next applies to both B cells and T cells and is a concept referred to as immunogenicity. Immunogenicity is the ability of an antigen to elicit an immune response. So what we can do then is discuss what makes an antigen a good antigen for an immune response to occur, or a strong immune response to occur. The first concept I want to focus on is what's referred to as foreignness. Foreignness just refers to a molecule that must be recognized as foreign. So the more unlike the self a molecule is, the more immunogenic it will tend to be. So antigens from more closely related organisms or species tend to be less immunogenic than compared to those types of antigens from very distantly related species. So the more phylogenetic distance between a pathogen and a host system trying to interact with an antigen is going to allow for more immunogenicity. Again, more phylogenetic distance, more divergence between the species is going to allow for a stronger immune response to take place. Now, one concept that's on this particular slide that we will eventually get to is a mechanism known as tolerance. Tolerance is an unresponsiveness to self-antigens. It is a built-in mechanism that allows for the protection of autoimmune situations. This is a mechanism built into the immune system to make sure that self components are not recognized as a potential threat. If tolerance mechanisms fail, that is when autoimmune disorders take place. There are two broad categories of tolerance, but again, we're not going to focus on that right now. I just want to highlight that there are two broad mechanisms for tolerance. When tolerance fails is when autoimmune disorders take place. But this is also really important in understanding, again, how distinct or different a potential antigen is from the host molecules will allow for a stronger immune response. Other features or characteristics that allow for a strong immune response to be elicited would include molecular size. There is a distinct and strong correlation between the size of a molecule of an antigen and its immunogenicity. So the rule of thumb tends to be bigger is better. Larger molecules definitely play a stronger role in eliciting an immune response. So macromolecules are, by definition, very large molecules, and hence macromolecules do in fact tend to make very good antigens. But that actually goes hand in hand with the next concept, which is chemical composition and complexity. The more complex a molecule is, the more immunogenic it will be. So, for example, with macromolecules, we know there are 4 macromolecule categories: proteins, carbohydrates, lipids, and nucleic acids. Proteins are extremely diverse. They are made up of 20 different amino acids and can range in size up to thousands of amino acids in length. So the size here is really important, as well as the complexity. Hopefully you all have had cell biology and have had some understanding of protein structure and know that protein structure is very complex. There is the primary sequence, or the primary structure, which is the sequence of amino acids present. The secondary structure that forms when alpha helices or beta pleated sheets form. The tertiary level is the folding or three-dimensional shape where amino acids interact to allow for this complex three-dimensional shape. And the quaternary structure is when two or more polypeptides can come together. So again, what this demonstrates is that proteins are very complex and have extreme chemical composition associated with them. So proteins tend to be the number one type of molecule that are extremely immunogenic. Carbohydrates can elicit an immune response, but are less effective. They typically have simpler structures, but can be more effective if they are linked to a protein. Same thing with lipids, in that lipids tend to be less immunogenic as well unless they are linked to a protein. And nucleic acids of the macromolecule categories are the least immunogenic because they are going back to distinction or foreignness. Nucleic acids are the same no matter what species we isolate them from. So the molecular complexity is very low and the foreignness is very low. Hence, nucleic acids do not tend to be very good immunogens. Now, this next slide focuses specifically on antigens that would be presented to T cells. And that is, processing and presentation is essential for that interaction between antigen and T cells. So the ability of a potential pathogen to be phagocytosed and processed is key to its immunogenicity. So typically things that are large, insoluble, and form aggregates make really good targets for phagocytosis. And if it's a good target for phagocytosis, will then eventually follow the pathway into a presentation to a corresponding T cell. Immunogenicity can also depend on a number of additional factors. Host factors are very important in eliciting an immune response. And when we talk about host factors, we mean things like the age of the individual. Typically, the very young and the very old do not have as strong of an immune response because the very young have not had a chance for that immune response to develop. The old tend to have that problem that we discussed with the thymus, known as thymic involution, where the thymus begins to shrink and not function at its optimum after the age of 65. So the age of the individual can play a role on how well an antigen acts to elicit an immune response. Disease states in the host can also play a role in immunogenicity. Stress, nutrition, fatigue all play a role in immune responses. And we probably are all familiar with the concept of what happens typically right around finals time is that we go. So we are stressed, we're probably malnourished, and we are also fatigued. And right after finals, what happens to a lot of people? We get sick. And that's because, again, our immune responses are a little bit lower and not being able to fight off that pathogen as easily. Genetic factors are also host factors that can play a role in immunogenicity. There are some specific genetic disorders that play a role in not allowing for strong immune responses to take place. We also have dosage that plays a role in immunogenicity. There's a critical level that needs to occur, a critical level of an antigen that needs to be present that will induce an immune response. If there is too high of a concentration of a given antigen, oftentimes that leads to tolerance. And this makes sense because something that is already in the host, a host cell factor, a host cell protein, for example, you would expect to have a higher concentration than, let's say, something that is a pathogen that has entered into the host system. So a higher concentration of an antigen usually does not elicit a strong immune response. Alternatively, very low concentrations can go under the level of detection and not be detected by the immune system. So here we have something sort of like the Goldilocks effect, where it's just the right concentration that typically elicits a stronger immune response. And then lastly, we have the route of entry. How exactly is this antigen entering into the host system? If it is ingested, for example, usually these pathogens get destroyed by the pH of the stomach. However, because of the route of ingestion, this is typically going to lead to something being less immunogenic. In comparison to, let's say, injection, like subcutaneous or intradermal, this is a direct entry of the pathogen into the host system and oftentimes does elicit a much stronger response. So how the pathogen gets in can also play a role in its immunogenicity. So we've looked at this slide once before in a previous slide. So this slide and the next slide really just highlights this concept once again. Again, for this slide, it's got B cell activation across the title, but the same mechanism occurs for T cells as well. What you see on the very left of the slide is that there are different populations of naive B cells. Ones that are represented by number 1, number 2, number 3, number 4, and are really nicely color-coded as well. These naive B cells have antibodies that bind to different antigens. So if an antigen comes along, in this case antigen number 2, that happens to have the ability to bind to the receptor, the membrane-associated antibody in B cell number 2, that then triggers the activation of that particular B cell and allows for production of lots and lots of B cells that have the same antigen binding capability. That is, they can bind to the exact same antigen that triggered their activation. So what you see here now is the activation and propagation of that specific B cell type to interact with that specific antigen. And in this case, we have memory cells that are being produced as well as effector cells. The effector cells for B cells are called plasma cells, and their job is to secrete soluble antibody that can then go and bind to that antigen that triggered the production in the first place. We will talk about what antibodies do in part 2 of this lecture. For now, let's just really get a good handle on understanding this activation process that takes place. This is just one more view of that same concept, in that in this particular figure, you have these B cells that are represented by the different letters and the different shapes associated with their antibodies, membrane-associated antibodies. And along comes antigen X that has the ability to bind to the antibody associated with B cell X. That then triggers its activation, which is known as clonal selection, and allows for the ramping up and the extreme production of that same type of B cell overall. This happens again for B cells as well as T cells. My goal is to always try and simplify this very complex concept associated with the immune system and the immune response. So oftentimes I simplify some of the concepts to hopefully help with your understanding. But as you will see throughout this rest of the semester, I always like to take it a step back and remind us of how complex everything is. And here is one of those examples. We know, like we just discussed, that with the naive B cell, there has to be that interaction with the antigen in order to start the activation process. It is a very complex process that occurs in that activation step. The antibody is in fact a receptor, and like we discussed in previous lectures, receptors bind to molecular signals known as ligands. So in this case, the ligand is an antigen that then triggers the activation and the differentiation of the B cell. But that receptor-ligand interaction that we talked about in very general terms applies here as well, but in much more complex level of detail. As you can see, there are a number of proteins that are associated in receiving this signal and causing the signal transduction pathway to take place. Eventually, what happens is several transcription factors are activated. Those transcription factors then allow for transcription of specific genes that allow for the differentiation of the distinct cell types, the memory cells as well as the effector cells. So again, not just magic when everything interacts, but a complex signal transduction mechanism does in fact take place. So now we'll focus a little bit more detail with antibodies and antibody structure. So antibody is a common, more slang-like term. The proper term for the molecular receptor is an immunoglobulin. So again, oftentimes we use the term antibody. However, the proper and correct term is immunoglobulin. On this slide, you can see several views of what antibodies look like, and we're going to talk about each one of these figures in a little more detail. Before we get into that, I want to highlight, though, for you that every antibody is composed of 4 distinct protein molecules that come together in a complex structure, quaternary structure. There are 2 chains that are referred to as heavy chains, and they are larger in size, and 2 chains that are referred to as light chains, and they are smaller in size. So in the bottom left figure is a very simplified view of both the membrane-associated antibody as well as the soluble antibody. What's highlighted here for you in the grayish blue color are the 2 heavy chains, and in the orange color, the 2 light chains. What holds all of these together are disulfide bridges that take place between different amino acids of the different chains. So there is a strong covalent bonding that occurs between these components to hold the structure all together. In addition, if we look at the top right-hand corner, there's a little bit more detail associated with these structures. What you can see from this figure is there are regions that are labeled with the letter V, and this stands for variable, and this is going to be portions of the antibody that can vary from antibody to antibody. This specifically is the portion that binds to an antigen, and this is what allows for that diversity and that specificity for distinct antigens. There are also those sections labeled with the letter C, which stand for constant regions, and these portions, the amino acid sequence varies very little from antibody to antibody. Going back to the figure in the bottom left-hand corner, what you can see is that these antibodies look almost identical, except for the portion, or the one that is membrane associated, has a longer portion that allows for that interaction with the membrane of the B cell. The soluble antibody does not have that portion associated with it. Going back to the figure in the top right-hand corner, you can also see that there is carbohydrate linkages that occur with the soluble antibody. This is what in fact allows for the solubility to take place, or to allow for these antibodies to be soluble. So the carbohydrate linkage is essential for that solubility. In the bottom right-hand corner, we have a more complex view of a typical antibody. And what I want to highlight here are the regions that are labeled FC as well as FAB or FAB. These were first identified when antibodies were first studied, and they actually broke apart upon extraction. And there were portions that were known as fragments that could crystallize, hence FC. And there were portions that were fragments that could bind antibodies, AB, hence the FAB portion. So the FAB portion is the portion where antibody interaction occurs with antigen. Specifically, it's the very tip of those portions that bind to antigen. And then the FC is what I typically refer to sort of as the stem of the Y. This portion is also extremely important for interaction that we will talk about a little bit later on down the road. So what exactly allows for the difference in the production of a membrane-associated antibody versus a soluble antibody? Like I mentioned in the previous slide, there is a section in the membrane-associated antibody that is absent in the soluble antibody that specifically allows for membrane association. In this particular slide, what you're looking at is a transcript, the result of transcription that takes place of a given antibody gene, or a gene that codes for an antibody. And we know that alternative splicing can lead to two different types of transcripts. One transcript that, when it is translated, has the extra portion of the protein that allows for membrane association, versus the form of the transcript that does not have that additional protein structure coded for in it. So this is the only difference between the antibody overall, is the splicing mechanism that allows for the addition of that protein section that binds to the plasma membrane, or omission of that overall.